So, yeah, good evening, everyone. Welcome to the Immunology live sessions. Today, I am Shimonti Aditya, a PhD, PMRF scholar in the Department of Biotechnology at IIT Madras. I am a teaching assistant for this course. Today, we will be looking upon the week 10 content of this course, and it's about cytokines and hypersensitivity and all those signaling pathways that leads to those. So let's begin without any delay. What are some terms we have been looking about in this last few classes? Those are cytokines, chemokines, interleukins, or ILs as we say them. So what are those, and what differentiates each of them? So cytokines, basically these are molecules. These are a group of molecules. All those signaling molecules that are, released by the immune cells, the macrophages, the dendritic cells, or these T cells, B cells. All those molecules come under this category as cytokines, and these molecules can communicate among different cells of the immune system and pass them different instructions, whether to increase the synthesis of certain genes, or whether to pass the signal that there is the site of infection there, so you need more number of cells to go and act in that location. So basically, it helps in cell movement from one part to another. It also helps in increasing the amount of cytokine production. The cytokines itself, acting upon different cells, will allow different cells to increase the amount of cytokines produced by them. So those ways, so these molecules are mostly soluble molecules, but some of them can exist as membrane bound forms. So soluble molecules, we have been talking about the complement proteins, right, in the last class. So complement proteins, these are inactive proteins, and they are cleaved into a soluble portion and an insoluble portion. So the soluble portion that it is cleaved into, those molecules can act like a cytokine and can induce signaling and also inflammation. So all these molecules are soluble molecules, and they are important for communication between the cells. What are chemokines? So chemokines, these are subpopulation of cytokines. They are inside cytokines, and they are subpopulation of cytokines, and they basically function in mobilizing the immune cells from one organ to another. So basically, it helps in the movement of cells. It helps in the movement of cells from one part of your body to another part of your body where the infection has been caused, or it can move the cell from one part of one organ to another part of that same organ. So chemokines basically function on just the movement of the cells, whereas cytokines can function in like signaling pathways. It can activate certain signaling pathways. It can activate inflammatory pathways. It can increase the amount of cytokines production from other cells. So everything cytokines can do. Chemokines are more or less concerned about the cell movement of the cell, or the cell migration. And what are interleukins? So interleukins are also a part of cytokines, and these interleukins were the first molecules that were discovered when when immunologists were studying how the immune cells mediate communication. So interleukins were the first molecules that were isolated, and they mediate communication between the white blood cells, the WBCs. And why the term comes as interleukins is, the term leukins comes from the word leukocytes, that means white blood cells, or WBCs, and the term inter stands for in between. So the communication in between the different leukocytes is mediated by interleukins. So that is about cytokines, chemokines, and interleukins. Like, how do cytokines act, and what is the mode of action of different cytokines? So there can be three modes of action. It is autocrine, paracrine, and endocrine. So what do you mean by autocrine mode of action? So here you can see, so molecules, cytokines which are released from the same cell, okay, it will be released from one cell, and it will go and bind to the receptors, the cytokine receptors, which are present on the same cell, and therefore signal the same cell to go on for different functions. And so this type of signaling is known as autocrane signaling. What do you mean by paracrine signaling? So paracrine signaling happens when cytokine molecules are released from one cell. This is one cell. They are releasing cytokine molecules, and these cytokine molecules go and binds to the receptor of a nearby cell or neighboring cells, and it will show the effect on this cell. So this cell will undergo any kind of signaling or any kind of movement or whatever downstream pathways. So cytokines released from one cell acting upon nearby cell. This mode of action is known as paracrine mode of action. What is the third mode of action is endocrine. So endocrine means the molecules are released from one cell into the circulation. So it is we know that there is endocrine glands. So endocrine glands also release their their secretions into the blood. They do not have any ducts for secreting any of their hormones. So rest of our hormones which are secreted by exocrine glands, they are secreted through a certain duct or a certain tube through which it is passed into their target organs. But endocrine mode of action means any of the molecules that are secreted by one of the cells, it is going to be released into the blood circulation. And from the circulation, because our blood can go and reach into different parts of your body, it can reach different organs. So from this circulation, the cytokine molecules can act on a distant cell which is very very far away from the cell which actually produce the cytokines. So endocrine mode of actions, they act upon cells which are located at a distance. So these are distant cells. Cytokine molecules are released into the circulation, and they act upon the cytokine receptors which are present on the cell surface of distant cells. So these are the three different modes of action of cytokines: autocrine, paracrine, and endocrine. So autocrine means it is the cytokines are released from the same cell, acting upon the same cell as well. Paracrine means molecules are released from one cell, acting upon the neighboring cell. And endocrine means molecules are released from one cell into the blood circulation, and it acts upon a very distant cell. So there are certain attributes of cytokines. There are certain terms which are associated with cytokines. Before that, we will look upon how cytokines work. So there is certain stimulus, inducing stimulus that will act on the cytokine producing cell. What could be the stimulus? The stimulus could be a pathogen entry into the blood, virus entry, or any kind of in your body, if there is any kind of scratch or any wound that is there, so that could be a stimulus. So that stimulus will allow the cytokine producing cell. So what are the cytokine producing cells? The WBCs, the macrophages, B cells, TH cells. So those are the cells which produces cytokines. And this cytokine producing cell, the gene which is particularly responsible for producing cytokine gets activated whenever there is a pathogen in the blood, or whenever there is a wound in your body. So or there is an inflammatory response is going to take place. So that time also there is a huge release of cytokine. We call it a cytokine storm during an inflammatory reaction. So those kind of stimulus, it will allow this cytokine gene to get activated, and it will release cytokine into the circulation, or into if it is going to act on a nearby cell, then into just it will release it in the tissue or in the extracellular matrix. So cytokines get secreted, and these cytokines will go and bind on the cytokine receptor present on its target cell, and this will particularly pass the signal. Cytokine binding to the cytokine receptor, shown here in gray, will pass the signal, and this will allow the gene activation. So what are the gene activations? So gene activation for movement of this particular cell to a particular location, or it might be proliferation of this cell. So if it is a B cell, then we need lot of B cells for producing a lot of antibodies if there is a pathogen against which antibodies are produced. So the signal will allow these B cells to proliferate and to grow in number, so that more amount of antibodies are produced. Then it will allow it to get differentiated. Some kind of T cells or T helper cells, due to the action of certain cytokines, it gets differentiated into different subtypes. So TH1, TH2, T helper follicular cells and all those. So it gets differentiated into different subtypes based on the different the cytokines that it is acted upon. So those kind of function, or it might also lead to cell death of the particular cell that is overly, expressing. If there is an inflammatory response, and if there is a huge production of cytokines, and it might go and act into this inflammatory, to reduce the inflammatory response, it might allow those cells to get killed. So those could be the effects of cytokines, and these are certain terms which are associated with cytokines. So we'll look into this. So these are pleiotropy, redundancy, synergy, antagonism, and cascade induction. So just before that, in brief, what do you mean by pleiotropy? So pleiotropy, it's a term which is used mostly. Most of the times we use this term pleiotropy, and pleiotropy means that if there is one particular cytokine, so say if there is IL-4, so this IL-4 can mediate different functions, and different functions can include activation and proliferation of the B cells. It can activate thymocytes. It can cause mast cell proliferation. So one cytokine, many function, that means pleiotropy. Okay. What do you mean by redundancy? Redundancy means there are different cytokines which are performing the same function. For example, IL-2, IL-4, and IL-5, they help in B cell proliferation. So IL-2, 4, and 5, they help in B cell proliferation. So their redundancy means that there are three different cytokines performing the same function. So that you mean by redundancy. What is synergy? Synergy means two cytokines together can perform one function. So either of the cytokine, when it is alone, it cannot perform that function. Only when the two cytokines come together, they can perform a particular function. For example, IL-4 and IL-5. So they can act upon by this two, IL-4 and IL-5 will act together and help in the class switching of the B cells. So again, what do you mean by class switching? So class switching is it happens during antibody classes being switched from IgM to different types of antibodies. So I had discussed that, okay, whenever there is an antibody secreted into the blood, or whenever there is a antibody which is membrane bound in the B cell, first type of antibody that is produced is IgM type, because the IgM gene is present in very vicinity where the cleavage of this particular gene occurs during this antibody production. So IgM then gets switched to different types of antibodies, IgA, IgE, IgG, etc. And in that that is known as class switching. And IL-4 and IL-5, the interleukins, these are helping together to carry out this class switching of B cells. What do you mean by antagonism? So antagonism means some kinds of cells, some kinds of interleukins, or some kinds of cytokines, they help in reducing the amount of any kind of effect. Like it could be class switching, or it could be like cell death, or it could be reducing the proliferation rate. So anything that is negative to what all other cytokines are doing. So cytokines basically help in increasing any kind of response, increasing the gene expression leading to cell differentiation, proliferation, etc. So any kind of interleukins that help in inhibiting that effect. Inhibitory interleukins or inhibitory cytokines are functioning as antagonists. So the term antagonists come from any molecules which inhibits any kind of reaction. So IL-4 and INF gamma, they help in inhibiting class switching. Okay, so INF gamma and IL-4, they help in inhibiting the class switching reaction, and they act as antagonists. Why is this necessary? Because all the time we need a balance to be maintained. So if a lot of class switching takes place, or if a lot of proliferation takes place, lot of differentiation takes place of the cells, then the balance will not be maintained, and there will be an overwhelming response from the immune system, and we do not want an overwhelming response. So those are the responses which can lead to autoimmunity or hypersensitivity. So these are the times when immune system do not function properly, or it is not maintained or not balanced properly. So we need certain cytokines which will which will show inhibitory effects. And the last attribute is the cascade induction. So cytokines basically it can act on any cell, and it can start a cascade of reactions. Like for example, complement proteins, it gets activated in 1, 2, 3, 4, 5 kind of fashion. So similarly, cytokines, one cytokine will go and activate another cell, and that cell will produce another set of cytokines. That cytokines will go and activate another kind of cell. So this continues, and ultimately the biological effect is achieved. So again, what do you mean by this pleiotropy, redundancy, synergy, and antagonism? It is shown here in this diagram very beautifully. So pleiotropy, as I said, that one particular cytokine can act as different, can show different functions. So for example, interleukin 4, it acts in the activation, proliferation, and differentiation of the B cell. It helps in the proliferation and differentiation of cytotoxic T cell precursor as well. And also it helps in the proliferation of mast cells. So all these three cells, it can act on all these three cells and perform the different functions in this cell. So this is known as pleiotropy. IL-4 is produced by activated T helper cells. Again, activated T helper cells can produce these three different kinds of cytokines, say IL-2, IL-4, IL-5. And redundancy means, see, each of these cytokines can have the same function. So IL-2, IL-4, and IL-5 individually can act on B cell and help in the proliferation of B cell. So even if IL-4 is not there, IL-2 can go and bind to the IL-2 receptors and induce proliferation. IL-4 individually can induce proliferation, IL-5 can individually induce proliferation, and so on. What is synergy is, there must be two cytokines which will act together as a team and will perform the function. So for example, here IL-4 and IL-5 together, see there is a plus sign, so they will act together on B cell and then it will induce class switching. So synergy and redundancy differs in a way that here in redundancy, one cytokine can individually perform the function, although different cytokines perform the same function. And here in synergy, both the cytokines, they need to be present together in order to perform their function and in order to carry on with the activities. And antagonism, again, antagonism is there are inhibitory cytokines which will block the activities. So for example, interleukin 4 and INF gamma. So interferon gamma and IL-4, so it will act on B cell and will block the class switching to IgE, which is induced by IL-4. So it is basically acting as an inhibitory cytokines. So see, IL-4 here can act as a proliferation signal as well. So IL-4 individually cannot act for class switching. IL-4 in presence of IL-5 can allow class switching, but individually IL-4 actually blocks class switching. So there are different functions of IL-4 as well, in individual and in and as a team. And cascade induction, so again this cell is particularly activated TH cell is so it is producing one type of cytokine, say interferon gamma, and this interferon gamma acts on macrophages or other kind of cells and can stimulate these macrophages to produce different kinds of cytokines like IL-12. And this IL-12 will act on, again, activated T helper cells and help in the production of another variety of cytokines. So there is a cascade induction where one particular cell is producing one kind of cytokines. This cytokines is stimulating another kind of cell to produce another kind of cytokines, and that cytokines again acts upon a different set of cells to produce the cytokines which will go and ultimately will act on the target cell and carry out the function. So there are different families of cytokines. So cytokines can be the interleukin 1 family, it could be the hemapoietin or class 1 cytokine family, it could be interferon or class 2 cytokine family, and it could be tumor necrosis family or interleukin 17 family and chemokines, etc. So all of these families, they were named based on the basis of the function that they do. So mostly on they have different roles in different parts of the immune system. So you can go through them. So I'll just read out. So interleukin 1, they comprises of this sorts of interleukins. You don't need to remember all of the names. You just need to know that what kind of classes perform what kind of function, and certain important interleukins like this which are performing Cleotropy redundancy. So certain examples like that is enough. Uh, then yeah, so interleukin 1 family, this is basically the first. IL-1 was the first non-interferon cytokine to be identified, and so interferons are kind of cytokines which are associated with viral immunity or viral protection against viral pathogens. And so IL-1 was the first non-interferon cytokine which was identified, because generally people identified this interferons and interleukins together, and the first non-interferon cytokine was IL-1. And members of this family can include important inflammatory mediators. So these are associated with inflammation. So interleukin 1 is associated with inflammation. Hemapoietin are class 1 family of small cytokine molecules, and it can show, exhibits the cycling sequence and functional diversity. And it also has different kinds of molecules like growth hormone, prolactin, erythropoietin or hematopoietin. So they have different functions. Growth hormone will help in increasing the growth. Prolactin helps in the milk production. Erythropoietin or hematopoietin, this also helps in erythropoiesis or red blood cell development, RBC development. And so interferon class 2 family, these are associated with antiviral responses. Interferons are always associated with antiviral response and protection against viral pathogen. All are important modulators of immune responses. So tumor necrosis factor, this family is associated with the immune system development, effector functions and homeostasis. So immune system, like you to build a better immunity, to build a better response against pathogen. TNF level should be quite maintained. TNF is associated with better kind of so all kind of stronger immune responses against any pathogen is provided by the TNF family of cytokines. And also the interleukin 17 family, this is the most recently discovered family, and these members will promote neutrophil accumulation and activation, and these are pro-inflammatory. So neutrophils again are the molecules which are the first molecules to reach at the site of action, action, and they can site of wound. So it can help in the accumulation and activation of neutrophils, and this can also be pro-inflammatory. Whereas chemokines, these are serving as chemoattractant function, cell migration basically. So yeah, so these are the different kinds of interleukins and different kinds of cytokines, different families of cytokines. And all these families, they are recognized by different kind of receptors which are present on the cell surface of the target cells. So all of this family of interleukins and interferons and cytokines and chemokines are actually recognized by different kinds of receptors. So they have different receptors and they perform different functions as well. So this is an IL-2 receptor. So this receptor basically functions in identifying the class 2, IL-2, IL-4 and IL-7 kind of IL-2, IL-4 and IL-7 class 1 family of cytokine is recognized by IL-2 receptors. So IL-2 receptors, they consist of an alpha subunit and a beta subunit, as well as what happens is the beta subunit, so the alpha subunit actually mediates the interaction between this the ligand and the receptor. So here you can see that there is an intermediate affinity because there is no alpha subunit. When there is an alpha subunit present, there is a low affinity. So alpha subunit helps in mediating interaction between the ligand and the receptor, and only this interaction will be supported, will be activated when there is a beta subunit bound to it. So alone the alpha subunit bound to the ligand, or alone the beta subunit and gamma subunit bound to the ligand cannot activate the responses. So all these three subunits have to be bound. So first the alpha subunit binds to the ligand, and secondly the beta subunit binds to this ligand associated alpha subunit and mediates the signaling. So basically in IL-2 receptor, alpha subunit helps in identifying the ligand and beta subunit helps in passing the signaling. So we will look about how the beta subunit has different kind of components in its cytoplasmic and its transmembrane domains that help in activating the signaling pathway, which will lead to different biological effects. So these are so basically again these high affinity receptors are those receptors which have both alpha beta subunit which are bound to the ligand, and this will mediate the signaling. Either alpha subunit bound to the ligand or the beta subunit bound to the ligand will not allow the signaling molecules to function properly. So this high affinity receptors are mostly high affinity receptor, and both low affinity receptors are present in activated CD4 plus and CD8 plus T cells. CD4 plus T cells and CD8 plus T cells are T helper cells and T cytotoxic cells, so all kinds of receptors are present on the cell surface, but mostly the receptors which are mostly not the receptors which have this kind of composition or this kind of combination, those receptors can only mediate signaling. Rest of these receptors will just bind to the ligand and will not mediate the signaling molecule, because the signaling is mediated by the beta subunit only when it is bound by the alpha subunit and the ligand. So again, this intermediate affinity is present on the NK cells or resting T cells in low numbers. So they do not mediate the signaling through the IL-2 receptors. So there might be some other receptors which are present on NK cells as well, which will mediate the cytokine signaling, but not this. So how does this particular interaction help in carrying out the signaling? So here there is an example of molecules which can act on the same which can act on the different alpha subunit but has a common beta subunit. So IL-3, IL-5 and GM-CSF or granulocyte macrophage, this colony stimulating factor, they will go and bind to its respective alpha subunits of the IL-2 receptor present on the cell surface of their target cells. So their target cells have this low affinity receptors. Basically, means the alpha subunit alone is low affinity. Low affinity will provide low affinity interaction. So IL-3, IL-5 and GM-CSF will go and bind to its respective alpha subunits, and once it is bound to its respective alpha subunits, the beta subunit will come and bind to this to the same alpha subunits, to the different alpha subunits of the different interleukin and the cytokine molecules. So one single beta subunit is there for one particular beta subunit can identify all the three different alpha subunits, and they will form an high affinity receptor. So now here there could be a problem, like if there is more IL-3 or IL-5 in your system, then IL-3 and IL-5 will bind to the alpha subunits more, and GM-CSF will bind less. So beta subunit that will be binding to the alpha subunits of IL-3 and IL-5 will be higher as compared to GM-CSF, because we have a limited number of beta subunit, right? We have a limited number of beta subunit, and we so mostly the beta subunit will be bound by these receptors. So this alpha subunit of this alpha subunit of the interleukin receptors, and so there will be less beta subunit present for GM-CSF, and this signaling will be reduced. Signaling which is mediated by GM-CSF will be reduced, whereas the signaling mediated by IL-3 and IL-5 will be in a exaggerated form. So that is why we can see that over here, when IL-3 and GM-CSF is binding to this particular alpha subunit, so there is higher number of IL-3 molecules. So IL-3 molecules will bind to this IL-3 alpha receptors, and GM-CSF is lower in number, so GM-CSF binds only to some of the alpha receptors. So we have a limited number of beta subunits. So beta subunits will go and bind to the alpha subunits of this IL-3 receptors, and there will be no beta subunits left for binding here. So the signaling will not be mediated, because the signaling is mediated by the beta subunit. Alpha subunit only mediates the interaction with the ligand. Okay. So here we will see how the signal is transmitted or transduced by class 1 and class 2 cytokine receptors. So yeah, there are different kind of receptors: class 1 receptors, class 2 receptors, class 3 receptors, based on the different kinds of cytokine families. So all of these cytokine families have different receptors. So we'll look into an important pathway of how class 1 and class 2 cytokine receptors is how signal is transmitted. So as I mentioned that alpha subunit and beta subunit has to come together in order to carry out the signaling pathway. So alpha subunit recognizes the ligand, and then the beta subunit comes and binds to this alpha subunit, which is recognizing the ligand. So there is a dimerization of the receptor. So alpha and beta subunit comes together. So once alpha beta subunit comes together, there is a cytoplasmic domain which is present in the beta subunit, and this beta subunit we call it as a JAK or Janus kinase. So this has a Janus kinase protein which is associated with the beta subunit of this beta subunit of this IL-2 or IL-1 receptor, and this will phosphorylate. Janus kinase is a kinase protein. Any kinase proteins, these are a family of tyrosine kinases. So any kinase is associated with phosphorylation. So kinase, the term kinase resembles any enzyme or any protein which is associated with phosphorylation. So Janus kinase will autophosphorylate all the tyrosine residues which is present in the beta subunit as well as present in the alpha subunit. So phosphorylation of this particular cytoplasmic region will bring about the docking. It will bring about the STAT molecules. STAT are another protein molecules. These protein molecules will be docked and brought about in this cytoplasmic domain. So what happens is, again, the ligand will bind to the alpha subunit. To that, the beta subunit will bind, and it will cause dimerization of the receptor. Once the receptor is dimerized, the cytoplasmic region has a JAK protein, that is a kinase protein. This protein will phosphorylate all the cytoplasmic tyrosine residues, and present in both the alpha subunit and the beta subunit. Now, this will carry out the docking, or it will send the signals for STAT molecule to come and bind over here. So STAT molecules will come and bind over here in the region of JAK, and JAK will also phosphorylate the tyrosine residues present in the STAT. So STAT molecules, initially they are present in the monomeric state. Once it is going and docking in the cytoplasmic region of the IL-2 or IL-1 receptor, it will get phosphorylated, and this phosphorylation will cause dimerization of STAT. So STAT will get dimerized, and dimerization of STAT will lead to its activation. So STAT molecules in its monomeric form cannot enter the nucleus. It remains in the cytoplasm. But once it gets dimerized, this dimerized STAT will enter the nucleus and will go and bind to the DNA and carry out this. It acts as a transcription factor. So what are transcription factors? Transcription factors are proteins which can go and activate a particular gene, or it can repress a particular gene depending upon what is its role. It can act as a promoter. It can go and bind to the promoter region and enhance the gene transcription, or it can repress the gene transcription. So STAT basically enhances the gene transcription of particular proteins which are associated with proliferation or differentiation or whatever functions this cytokine wants to mediate. So that is the pathway. So JAK gets phosphorylated, then it phosphorylates STAT. STAT then gets dimerized, and then dimerized STAT will enter the nucleus, go and bind to the DNA, and carry out specific gene transcription. So now we will look into certain aspects of hypersensitivity reactions. So what is hypersensitivity? So hypersensitivity in our general term, in our general like general way, we call it as an allergic response. So allergic response can happen to lot of people, and it is associated with your immune system not acting properly. So if your immune system acts in a over dramatic way, then you will get this kind of allergic reaction. And it's very common in spring, and in this when pollen grains are, pollen grains are in the air, so people get sneezes, and people cough. So all of those reactions are associated with hypersensitivity reactions or allergic reactions. And those allergic reactions could be again of four types, depending upon what is causing the reaction. So type 1 allergic reaction is mediated by IgE type of antibody. Type 2 is mediated by an IgG or IgM. It's an antibody mediated hypersensitivity. Type 3 is mediated by immune complexes. So immune complexes, those are present in your circulation and it is not removed from your circulation. So those immune complexes will create a problem. And type 4 or delayed type hypersensitivity is caused by T cells. So type 1, type 2 and type 3, these are associated with antibodies. Okay, antibody or antibody antigen complexes. And type 4 is associated with T cells. So what happens is, so here we can see in type 1 kind of antibody, there is an allergen. So this allergen, these are basically presented by certain antigen presenting cells through the MHC class 2 molecules. And this MHC class 2 molecules will go and prime with the B cell, and allow the B cell to recognize this particular allergen. And during that B cell recognition of this particular allergen, and class switching of this allergen, there will be the antigen specific or allergen specific IgE molecule will be produced. So this IgE molecule will lead to the hypersensitivity reaction. And what happens is, in this IgE molecules, it will go and bind to different mast cells. It will go and bind to basophils, and IgE molecules will basically bind to basophils and mast cells and cause degranulation. So degranulation means it is releasing all the granules of histamine and prostaglandin and leukotrienes. So all these molecules are associated with inflammatory responses and allergic responses. And so IgE will mediate this kind of responses. So again, allergen is binding to your molecules. So allergen, it is engulfed by any macrophages or B cells or neutrophils, dendritic cells. It is processed and it is presented by MHC class 2 to your B cells, and B cells will recognize this antigen or allergen and then will produce antibody, which is IgE type, allergen specific antibody, and will this IgE type will mediate the hypersensitivity reaction. And IgE will go and bind to mast cells and basophils and release these vasoactive mediators. So antigen will induce this cross-linking of IgE bound to the mast cell. So yeah, cross-linking is important. So this IgE molecules, when bound to the allergen, if it is bound individually, it is not going to produce any inflammatory response. If it is bound in a cross-linking way, so same antigen is bound to two different IgE molecules, so it will cause a cross-linking of that, and that will release in the degranulation. So what is the typical manifestation? Typical manifestation that will induce anaphylaxis. And what do you mean by anaphylaxis? Anaphylactic shock is when you are not able to breathe properly, as you're asthmatic, you're having asthmatic shocks or asthmatic attacks. And there could be food allergies or eczema. Eczema is a skin allergy where you get red rashes or spots in your skin. So those kind of responses you get in the type 1 allergic. Type 1 allergic is the most common type of allergy that people experience, and it is due to IgE production. The second type of hypersensitivity reaction is the type 2, or the antibody mediated hypersensitivity reaction. What happens is, antibody will direct the cell surface antigens, and it will mediate the destruction via the complement activation, or ADCC. ADCC means ADCC is I forgot it. I'll let you know. So yeah, so what will happen is, antibody, this will be, so your cells which are bound to your the molecules, the cells which are bound to your antigen presenting cells, this will be presented by MHC class 2, and MHC class 2 will then go and bind to your B cells, which will have these antibody molecules, and these antibody molecules will bind to this antigen presented by the MHC class 2, and this will form an immune complex, antigen antibody complex. This antigen antibody complex will then activate the complement proteins which are present in your blood, and this complement proteins will go and bind to this FC region, and this FC region will result in a greater cleavage of your complement proteins. So this complement proteins have a soluble portion, that is the C3A or the C4A, C5A, all those A portion, these are the soluble portion, and this will help in the complement activation. Those complement activated proteins will then go and increase, will cause a greater immune response, and this immune response will is known as the type 2 hypersensitivity reaction. So this happens when there is blood transfusion reaction or erythroblastosis fetalis. Basically, whenever there is a erythroblastosis fetalis or blood transfusion reaction happens, whenever there is mismatch of your and mismatch of your and specific like mismatch of your certain proteins present in the, the donor and the receptor. So in erythroblastosis fetalis, there is like the mother is Rh negative and the fetus is Rh positive. So during the delivery, when there is mixing of blood, there will be a mixing of blood. So your mother's body that does not have an Rh antigen, it will generate antibodies against the Rh molecule, and so during second pregnancy, if the baby is again Rh positive, so the baby's blood will be rejected by the mother's blood through the IgG mediated immune response, because IgG can cross the placenta. So that way, this is known as erythroblastosis fetalis. It could be very deleterious for the fetus. And blood transfusion reactions, where the blood type is not matched, whether your A individual is getting blood from the B individual, so all those antigens, they come and play, they come into effect, and they will cause complement activation. So this is type 2 mediated hypersensitivity. Type 3 mediated hypersensitivity takes place when there is an antigen antibody complexes which are present in your system and it is not removed properly. So antigen antibody complexes need to be neutralized. It need to be phagocytosed, or it need to be removed from the circulation. If it is present in the circulation for a longer duration of time, then there will be then that particular complex will cause again complement activation, which will result in the inflammatory responses through the infiltration of neutrophils and mast cells and basophils. So this will result in the immune complex mediated hypersensitivity or type 3 hypersensitivity. It happens due to antigen antibody complexes which are deposited in different tissues, which will induce complement activation, and again inflammatory response mediated by the infiltration of neutrophils. This includes localized Arthur's reaction and generalized reaction, such as serum sickness or necrotizing vascularitis, rheumatoid arthritis, etc. And type 4 hypersensitivity is the hypersensitivity reaction which is mediated by the T cells alone, where what happens is the T cells which are priming with your B cells or an activated macrophages, this will release a higher amount of cytokines in the circulation, and this kind of cytokines will again go and act on this macrophages and will cause tissue damage or cell death. So this cytokines, a huge release of cytokines or cytokine storm, will result in the cell death or cell tissue damage, and that will result in a type 4 kind of hypersensitivity reaction. This will include dermatitis or tubercular lesions and graft rejection, etc. So all those responses that requires adaptive immune system, those kind of responses are those kind of hypersensitivity reaction are mediated by type 4. So, what are the mechanism for the type 1 hypersensitivity reaction? So, there is a B cell, and this B cell is having an allergen which is bound to some antigen antibody complexes which are IgE type, and this allergen is this is a cross linking of IgE type of B cells. So this allergen is also processed and presented by MHC class 2 to the T helper cells, where T helper cells are interacting with this allergen, and it is producing a lot of cytokines. So what happens whenever there is a lot of cytokines production, there will be B cells which will be proliferated into memory cells and plasma cells, a lot of memory cells and a lot of plasma cells. So these plasma cells will again produce a huge amount of IgE antibody, which is again specific to this antigen, and this IgE antibody will have certain kind of Fc receptors present on the sensitized mast cell, and this IgE antibody will go and bind to these Fc receptors present on the mast cell, and once when already the allergen is there in the blood, so when allergen will bind to this IgE which is already bound to the mast cell of mast cells, this IgE will show cross-linking. So these IgE molecules, these are separated. When allergen binds to it, it will show cross-linking, and this cross-linking will result in the degranulation or release of certain kinds of molecules like histamine, heparin, proteases, and prostaglandins. Those are those molecules. These are associated with different kinds of effector functions like pain or inflammation, redness, swelling, etc. So these are vasoactive amines which are released from the circulation, and these are smooth muscle cells, small blood vessel, mucus gland, blood. It can act on different kind of cells. So all of these vasoactive amines can act on different kinds of cell and can carry out different functions like pain or proliferation or cell death, etc. So that is all. Now we'll discuss some questions. So you can just answer me this in the chat window. So it's not C. I actually mentioned that the alpha subunit binds to the ligand and beta subunit mediates the signaling. So the option is option number. The correct option is option number B. Because alpha subunit binds to the ligand and the beta subunit is transducing the signal by activating the JAK kinase, and all these JAK-STAT pathway is activated. So alpha subunit does not transduce the signal. It is the beta subunit that transduces the signal. Okay. So let's see the next subunit. What do you mean by redundancy? And what is redundancy? Right. Option is the correct option is answer number B. Uh, because the same alpha subunit will not bind to the different kind of receptors. Alpha subunits are different for each of these ligands, because the ligands are bound by the alpha subunit, but the signal is transduced by the beta subunit. So the same beta subunit transmits the signal, but the alpha subunit is binding to the different cytokines. So the correct option is answer number B. So this was discussed in the video. If you have seen the NPTEL video, what is the correct answer for this? So this was discussed in in lecture number 47 of the NPTEL video. It's not option number A. It's option number D. It is basically IL-6 will help in T follicular helper cells development. TH1 is help. TH1 development is brought about by IL-12 cytokine. TH2 is by IL-4. TH17 is by IL-6 and IGF beta, and T follicular helper cell is by IL-6. So IL-6 helps in the development of T follicular helper cells. So yes, this was also discussed in the lecture. So STAT basically goes and binds to the DNA region, and in the DNA region it will bind to a particular segment. So that segment is known as some what is the name of the segment? It was also discussed uh, in the class. Right. It's option number A. This is interferon sensitive response element, or ISRD. So that is the region where STAT dimer goes and binds in the DNA and will help in the gene expression of the particular gene, whatever the cytokine is wanting to regulate. So STAT dimer will bind to ISRD region. That's right. And this is the last question for today. So this was again discussed in which lecture? I'll tell you. Lecture number 48. So this particular part of the immune system is little. It requires a little mugging up. It requires little memory based questions, because which interleukins is going to activate which T helper cell, and which cells are producing which interleukins or cytokines. And there are certain kinds of cytokines which are associated with like this IL-4, IL-5 or IL-6. So these are some kind of cytokines which are important, and their activation and their synthesis as well as their role is kind of important in knowing. For example, also this interferons and TNF, certain family of certain cytokines of TNF family. So all these are important for knowing, because these are like some memory based questions always come up from this topic, and nothing to do, we have to mug up that, or you can write it down and then just go through if you do not remember it. So yeah, so this question, oligoadenylate synthase will activate the RNase L by what kind of reaction? So what is oligoadenylate, you know, synthase. So these are again activating. These are associated with interferons, associated with the interferon receptor or interferon stimulating genes, and this oligoadenylate cyclase along with MGTPase and PKR or protein kinase R, they belong to the interferon stimulating genes which are regulated. And how are they regulating the viral proteins, and how are they providing viral immunity? So that is discussed again in lecture number 48. I will just go through briefly. So OAS, they make a 2 prime to 5 prime adenylate cyclase phosphodiester bond. So the correct option is oligoadenylate 2 prime to 5 prime uh, uh, for this thing, oligoadenylate bond 2 prime to 5 prime phosphodiester linkage between oligoadenylates. And what it does is, instead of like whenever there is a monomeric form of OAS present, it will go and bind to the viral mRNA or the viral DNA or the viral RNA. So, and then this OAS will form the OAS tetramer. Once it binds to this viral RNA or DNA, the genetic material of the virus, it will form a tetramer, and that will initiate the synthesis of 2 prime to 5 prime oligoadenylate or oligoadenylate phosphodiester bonds. And this phosphodiester bond or this phosphodiester linkages will interact with another kind of protein which is known as RNase L, and RNase L helps in the cleavage of the viral RNA. So basically OS binds to the viral mRNA and then forms a tetramer around it. This tetramer will form 2 prime to 5 prime oligoadenylate bonds. This bonds will help in binding to the RNase L. RNase L will form a dimer, and that dimer will be cleaving the viral RNA. So that way, this interferon stimulating gene OAS, it works and it carries out its function in providing viral immunity. And so that is it for today.